Hello and welcome to the recording of Season 1, Episode 9 of Proteus TV. My name is Julie Hayes. I am a PhD student in computer science here at the University of New Mexico. And this episode had been an interview with two University of New Mexico scientists, Professor Judy Cannon, who is the professor of microbiology and molecular biology, and she's an immunologist here at our Health Sciences Center, and Professor Melanie Moses, who is a professor of computer science and biology here in our engineering department. And they have been longtime collaborators, and we at Proteus are interested in lots of kinds of scientific collaboration, especially among computer scientists and biologists. And so this is an interview about their longtime collaboration, how they got to be collaborators, how they're interested in the same questions, and some of their work together. I forgot to record this episode as it was being recorded. Well, I didn't completely forget. I remembered halfway through. So I will be splicing you in to the interview in progress, the recording of the interview in progress. And Professor Cannon is talking about, she's answering the question of how the two of them got to be working together and the sort of interests that they shared. And if you're wondering what her background is, it is the Rio Grande that runs through Albuquerque, and it's very beautiful. And nobody knows this. It's a big secret that Albuquerque is beautiful. Welcome and enjoy the interview.
Target cell, in this case, in general, is a virally infected cell, but it could be a tumor cell. So a lot of probably the most novel and efficacious new cancer therapy is to use this capacity for T cells to directly kill tumor cells. But in order to kill, they do have to make direct physical contact, whereas a lot of other mediators of our immune response don't have to touch. They release factors that can go through our bloodstream and float around and do their function that way. And so T cells are particularly interesting because they have to physically move in lots of different spaces. So one of the questions we've been interested in is, what do they do in all these different areas that they move to? So we mostly look at the lung, and in some cases the lymph node, because that's where actually T cells start off. And that we have lots of nodules all through our bodies. That's where T cells, when there's no infection, they sit there. But once they sense infection, they get activated in the lymph node, and then they have to go to where the infection is. So if we get SARS, they start in the lymph node that's near the lung, and then they go back to the lung in order to kill off. And so how does it move in the lymph node? Because the lymph node looks totally different from the lung. There's lots of different cell types, there's lots of different proteins, and the structure itself is totally different. And so, how does a T cell know how to move in all these different places? Do they move the same way? Do they not? And so that, I would say the biological approach that I had done mostly is we do a lot of molecules, like we think about factors, proteins, and so we can knock them out is what we do. We have used animal models, and so we can make animals that are deficient in one molecule or another. That's really where I started, and we have theories, hypotheses about what certain molecules or classes of molecules do. So we can ask, yes, if the T cell doesn't have this molecule, now it doesn't move as fast, or it doesn't move, to these tissues as well. So, which is very interesting, and that's really the biologist's view. But when I started talking to Melanie and she started talking about patterns, I was like, what do you mean? This is just like completely foreign concept. Like, either they go there or they don't, or they move fast or they don't. For us, it's a very binary often. It's like, do they die or do they live? It's like we're not very finely tuned to the, sort of these bigger kind of potential questions. And so that's where when Melanie said, with ants, we know that they can do these kinds of movement, and that actually can matter. And I was like, actually, I never ever thought about that before, and that makes a lot of sense to me. Also at this time, actually, a paper did come out where another group had collaborated with actually a biophysics group and started thinking a little bit more about these questions too. So again, I think, we weren't necessarily the only ones, but once we started talking, it's like, oh, yeah, hey, with these, and then somebody else had started applying to T cells. So it really did seem like, oh, yeah, this is absolutely a fruitful, way we should start thinking about this, actually. And So I would say, Melanie brought in just a lot of like patterns of motility, as well as thinking about it in terms of search efficiency, not just do they get there or don't they, right? Like, can they do it quicker or slower? And could that matter? Like, it's not that they just live or die, but if they got there a little bit faster, would that be better? And actually, I feel like this is partly why we've been so interested in SARS, and SARS has been a very interesting test case here, is that we do think a little bit more, a little bit faster might make a lot of difference, actually, right? It's not that it might not be, oh, you have no T cells or you have a lot of T cells. It might be, well, if you had a few more T cells or a few fewer T cells, and they move in certain ways, like the difference, the whole range of responses in a disease like SARS, where some people barely get sick, other people get sick, other people have to be hospitalized, other people die, right? There's a whole host of, like, a range of responses that could be, more dependent on these kinds of questions that we've been asking, rather than, right, you're totally deficient in something. Now every mouse that's deficient in that dies. Like, that's not really how biology generally works.
Yeah, so it was not necessarily the computational biology, which is something you've done a lot of, but more the biological computation lens of the system that really offered a different way to answer the questions that you had.
Right, and I would say it was not about, oh, here's a new tool, let's apply it. It was always, I feel like, and Melanie, I don't know if you would agree with this, but that it was always about the questions. Like, oh, here's a really cool thing. Like, what tools can we now use? Oh, yeah, computation is a great tool, actually, right? And so I feel like it wasn't, oh, you're a computer scientist, let's use computer science. Because I do feel like that's more and more people are like, machine learning is so cool, let's use machine learning. Like, on what? You know what I mean? Like, what do you want to do with it? I mean, sure, it's a cool tool, but I feel like, for me, it was still like, okay, what are our questions? And then, oh, yes, I mean, competition is a great way to approach it. Hardly anybody's done it. I certainly don't have the tools. And so, but it was not really about like, oh, computer science is cool, let's use it. It was more, okay, yeah, this is a search question, and you have a set of tools I don't have access to. And then, but we could provide some, I think, data, a set of tools too for Melanie that she had not had access to also. And so, then it could feed back and forth.
Yeah, I'll chime in. So I completely agree. And, I think the vast majority of work in computational biology is what Judy said, that there's a biologist who has a problem and they look to computer scientists to give them tools to solve it. Right? I have this very narrowly, focused question, and there's some machine learning approach that will answer it. And I think what we've been doing, I hope, is representative of what we can do at the Proteus Institute, which is this sort of reframing of the question, using both a computational lens, but maybe even more importantly, this kind of complex systems lens. And the immune system, to me, I'm going to stand by the statement. Some people disagree. Everyone has their favorite complex system, but I think the immune system, the adaptive immune system in particular, is the quintessential complex adaptive system. I mean, it's super complex in that there are many different kinds of agents or cell types. They do different things in different places. Their communication is really important. Your health is really just this emergent property, right, of how all of these different cells interact with any kind of invading pathogen and the rest of the cells in your body. It's just it's almost too complex, right? It's which is some of the trick, is figuring out how to simplify enough to make progress and study it. But it just really, and T cells in particular, I think exemplifies some of the things that I've been interested in complex systems. So search problems to me are interesting, although I'm interested in them mostly because I want to understand how cooperation can work. And T cells are an example where the cooperation is very emergent. Like, there's not a lot of examples of T cells doing something that you would sort of point to that, and that you would say, this is where the T cells are cooperating with each other, right? I mean, there's some signaling and communication, but it's not quite as obviously cooperative as an ant colony, right? Where it's clear that all of the cells are working together toward this common goal, say, working for food. It's much more, I think, dependent, the behavior of the T cells, as Judy was saying, is really dependent on, for example, the structure of the tissue that they find themselves in. And that was actually an argument we made in a paper that that the way that T cells respond to their environment is where their environment is a lung or a lymph node or a liver. They need to have rules that are sort of generalist rules that become very particular for the environment that they're in, which is something I think is quite analogous to the way that individual ants, have sort of a generic set of rules about the way they interact and they pheromone trails and communicate to each other. And that plays out very differently. Sort of there's evolved strategies that play out differently in different environments by different groups of ants. What's cool about the T cells, though, it's the same T cells, right? The T cell doesn't know from the start. Right? Evolution hasn't told it, when the T cell is created, that you're going to be a T cell that ends up in the liver or the lymph node. Right? So there's this extra level of kind of flexibility and adaptation there that is just completely fascinating to me. As Judy said, just the more I learn, the more I'm amazed any of us survive any of these viral infections, right? Because the process that the immune system uses to fight it is so complex but complicated. Like no engineer would ever build a system that worked this way. Right? It's a great example of how evolution creates, this sort of entwined, complex system of cells that even though it's very hard for us to make sense out of what they're doing, right? Many thousands of immunologists are spending a lot of time to figure out what each of these cells does in each location in different conditions. It works surprisingly well. So it's been exciting to address those kinds of questions, both from computational tools where we can build models. We have individual agents, individual T cells moving around in the space of the lung, but also Judy can, do an experiment with a mouse where we can watch real T cells and how they respond in these different environments. And that's, I think, the iteration between those two approaches is what's really powerful.
Yeah, so for the folks at home who are a little confused, we'll start with Melanie. What is a complex adaptive system and what is an emergent property?
Okay, great. So that's the words we toss around like everybody knows when we mean this. So a complex adaptive system, to me, is any system, most of them are biological, but some are sort of social systems. Any system with a set of individual agents that interact with each other, and the product, the result of their interaction, is something that's kind of non-linear. What we say is more than the sum of the parts. So you don't actually understand how the immune system works by just understanding, for example, what an individual T cell does. You have to understand the way that it interacts with other T cells, with other features of the system. And then there's some emergent property, right? Something that results from those interactions that's maybe not easily predicted from knowing the rules of the interaction itself. And emergent is a word, for a while it's kind of a dirty word. It got these sort of spiritual kinds of connotations to it, and people thought it wasn't grounded or scientific. But it's a very simple concept, and I think a really useful one. To me, one of the ways to think about it is that temperature is not a fundamental property of like a single molecule, right? You have H2O, a single H2O molecule. It doesn't have a temperature. It needs to interact with other things in order to have temperature. Temperature is measuring how quickly it's bouncing off of other molecules. So it's a property that emerges when you have lots of individual agents that are interacting. That the speed of how things bump into each other requires that you have multiple instances of them. So there are lots of these emergent properties that get to be more complicated and harder to predict than maybe temperature of molecules bouncing around in the room. But that concept, that framework, that you don't actually understand how a system works by just knowing the rules of how every single thing in the system works. You essentially have to understand, in many of these the interesting cases, you have to understand, generally, you often have to simulate the interactions in order to understand what will happen under different scenarios. The combinatorics of the possibilities of the ways that things interact differently are huge, and so you need these kind of computational skills, tools to make sense of the system.
Yeah, so for the folks at home, for this is new too, we take a system of inquiry and we abstract the parts of the system out into what we call agents, be those molecules that collectively have an emergent property temperature and that singularly have behavior, but we can't understand it until we see collectively a behavior of search, or T cells that collectively have seemingly random behavior, but together have the incredible ability to fight off diseases in our body, including novel diseases like the coronavirus, which I'd like to go into a bit more. That your work on SARS and your unique take in understanding and modeling the emergent behavior of this virus and our immune system's adaptation to this virus. So I'm not really sure where to start here. This is the most recent thing after ants and T cells. This is the newest phase of this collaboration, but I think either one of you will have a good sense of where to start the story of SimCove.
Should I can start, and Judy can correct me when I say something immunologically incorrect.
Well, and actually, let's start with what were the questions that you had been asking and or answering that brought you into this this phase of the inquiry.
Great. So Judy and I and Stephanie Forrest and a few others have been collaborating previously before SARS-CoV-2 came on the scene. We've been modeling influenza in the lung, and we had taken a complex systems approach to understanding the T cell response to the spread of influenza abstractly in the lung, or influenza infection, maybe even a better way to put that. And so in that work, we used a technique called agent-based modeling, which is what I've done for a while and what we are continuing to do with SARS-CoV-2. And the idea there is that instead of just modeling how a disease spreads using differential equations, which is kind of the typical way that disease is modeled both within your body, and now people are very familiar with the idea of epidemic modeling, right? When you model, how many people does any individual infect and how many do they infect, and right? And you can model that with an equation and a set of kind of coupled equations that relate the ability to fight a disease, the immunity, that some people or cells become immune over time, and all of those interactions can be modeled mathematically. We took the approach of modeling that as spatial interactions. And one of the reasons is that Judy pointed to earlier, for a T cell, for example, to kill a virally infected cell, it has to be right there, right? This is not something you can do at a distance. And so spatial interactions become really important in understanding how virally infected cells are killed. And so we did that explicitly. So we made essentially little, it's almost like a little video game showing, one cell is infected, it infects its neighbors in space. The infection grows. Over time, you have T cells and B cells that are adapting, or that are the population of them. A small number will recognize that pathogen. That population will grow, and then those, in this case, T cells, will go to the site of infection. They get attracted by chemical signals, and we can watch the dynamics of the T cells killing the virally infected cells. And one of the things that came out of that is that the spatial distribution really does have an effect on the dynamics of the virus that aren't captured in abstract mathematical models. And so this was in some ways just sort of an interesting curiosity for us with the flu, because those spatial effects probably weren't usually that important. But what we saw when SARS-CoV-2 infections started to be shown, people would show CT scans or x-rays of the lung. And one of the things that was really shocking and is still kind of horrifying to me is how much infection there can be in someone's lung from early in the infection, which is generally different from the flu, and how that infection can spread quite rapidly. And so our first big insight here was that it looks like the amount of virus you have in your lung, according to our modeling, is really highly dependent on how many different locations got seeded with infection from the start. And that the more essentially virus particles you breathe down into the fractal branching structure of your lung, the more disparate locations are infected, the more there's possibility for spread to happen in space and keep encountering new uninfected cells. And so that was sort of the big one of the big insights, that it's not exactly just how much virus you inhale, it's how much virus you inhale into different locations. And that has a lot of potentially to say about aerosols and how they spread in your lung. And so we've been somewhat focused on that question. But then the T cell response is also dependent on that spatial distribution, because T cells go to places where there are these signals, these inflammatory signals that say there's an infection here. And so we can actually spatially kind of play out the dynamics of the T cells attacking those locations. And as Judy alluded to before, having a sufficient, there are sort of thresholds. If you don't have quite enough T cells quite early enough, the infection can get out of control. And so it could be that a large part of the differentiation between people is, who happens to have a strong T cell response. We know some of the reasons for that, right? Older people, particularly people over 65, tend not to have a great immune response to a novel pathogen. Right? So they might have delayed or largely reduced T cell responses. So we can actually just sort of play out these dynamics, hypothesize how they work in a computational model, which is a whole lot faster and cheaper than doing this in a petri dish or in a lab animal. And so that's the work that we've been working on over the last, I guess, year and a half at this point. And hopefully is coming out soon.
Wow, yeah.
From my side, the influenza, I use influenza in my lab, but I would say almost as a model, we know that T cells respond in both humans and animals to influenza, but the real protection probably comes from the B cells, which I haven't hardly talked about because they don't work on them so much, but that's another arm of the immune response. They make these molecules called antibodies, which are critical, especially for protection, say, after vaccination. And in many infections, antibodies can also play a really important role. But depending on your infection, this is why we have so many different, what we call effector arms of the immune response. Like some, you need one kind of cell, some you need another kind of immune cell, right? Because we encounter such a huge variety of pathogens. And so that for the flu, a lot of work has gone into understanding the antibody response. Whereas it's interesting because SARS, it's similar. It's a virus. Of course, it's a different kind of virus than influenza, but that it's respiratory. But it seems to be much more dependent on the T cell response than, especially in a primary infection, as we call it. Like if you just get exposed to SARS and you get it. Whereas the vaccine induces an incredibly strong antibody response as well as a T cell response. And so you can have both arms of protection if you happen to encounter SARS after vaccine. But if you just encounter it the first time as a live virus, that the T cells seem like they play a really crucial role in clearing. So people who have deficient T cell responses seem like they do worse, or sometimes the T cells get out of control, actually, and then you do worse as well. So again, T cells, too little is bad, too much is bad too. So there were a lot of interesting aspects about the T cell response with SARS. So as this data was coming out to me, it seemed like, yes, we built the model for flu, and we could easily, turn it to SARS because a lot of aspects of it were similar. But I thought, as a T cell immunologist, it was even more interesting because the role of T cells seemed to be even more relevant here.
And Judy, that spatial model, I imagine that's pretty unique as well among immunologists. Is that what has been the reaction to that, and your thoughts about that in the immunological community?
Yeah, so I would say that modeling for immunology is still a little bit behind. I would say a lot of other, Melanie mentioned, especially with SARS, epidemiological modeling is very common for infectious disease. But I would say that other aspects of modeling, which I would say in cancer, in certain fields, are much more not only widely accepted but utilized, and a lot of biologists look to modeling. But immunology, it's still, there's certainly people who are interested, but I would say that and Melanie mentioned there's different kinds of modeling. Spatial modeling is even a little bit, I think, not as sort of mature, advanced, not as many people do it, but right. And I would say that maybe it's not as relevant, again, except for T cells and certain kinds of cell types, right? I think we've all, Melanie always says things like this, which is like every model has, what is it? Every model has problems, but only some are useful. I can't remember exactly what.
All models are wrong, some are useful.
Exactly.
Very famous statistical quote, but it applies to all kinds of models, including the ones we have in our heads.
Exactly. So that, I do think that it's been nice. Like you said, we developed a spatial model, and actually we published a paper for flu saying that, well, maybe actually T cell responses aren't so important there, actually, right? But that for SARS, it is more important. So then it is more, relevant to build a spatial model for these kinds of parameters in these kinds of cases for the T cell response. So I feel like this next phase I'm very excited about, because I do think that rather than, we have this tool, let's just use it for everything, whether, and sometimes we do that, right? Like it's sort of like, oh, we have it, why not? But that I would say now it feels particularly relevant.
Yeah, and for the folks watching at home, when they're saying SARS, these two are saying SARS, they mean SARS-CoV-2, the virus that causes coronavirus, or COVID-19, or COVID, as we all are familiar.
Yes, exactly, which causes COVID-19.
Not the first SARS. Awesome. Well, as professors, there are many demands on these folks' time, big on ideas, short on time. So as a wrap-up, I'd love for each of you, we'll start with, what advice would you give to scientists watching this that would like to be more collaborative or interdisciplinary?
Maybe I'll start. I think that you have to be willing to compare it to T cells. Like, it's like, I don't know, Tinder dating nowadays, whatever, where, and you do have to talk to a lot of people, and many of them will not, right, whether the science is not a great fit, the personality, because for really productive collaborations, it's definitely beyond scientific interest. Like you kind of have to sort of match styles. It's like finding a life partner, maybe, where you do have to sort of match on several levels, and that it's really lucky and productive when you do. But it does, I feel like, sometimes take, you can't assume, oh, this person should fit, let's talk to that person, and like sometimes I feel like it's a little bit more random, like, yeah, let's talk to a bunch of people and hopefully one will really stick. Maybe none will, but if you're not willing to be open to really listening to different ideas, then I think it's it's less likely. But if you are, then the possibility is there for sure. Sometimes it still doesn't work out, but then it could.
So make sure you have a large sample size when science tindering. Melanie.
Great. Yeah, absolutely. Agree with Judy 100%. I think there's another lesson from the immune system, and that is sort of being willing to fail. Like, it's a similar idea. Like, try a lot. Most T cells are totally useless. The vast majority of your trillions of T cells you have over your lifetime will do nothing. And so it's the population of T cells that are trial and error, and every now and then one of them is going to save your life, right? So I think that's an important piece of this. Even any evolutionary process, it only works if things die, right? I mean, most things have to not work, and so these kinds of collaborations that are kind of trying to move into new areas that kind of exist between disciplines. There's a huge number of those areas that are really interesting, but probably a bigger number of those areas that either aren't interesting or we just don't quite have the tools to solve the problems in those areas. So, this is a place where exploring and playing and having a lot of fun in the process, asking interesting questions and just seeing which ones work, I think is super important. And right, finding somebody that you work with who has a similar tolerance for maybe failure and experimentation is probably an important part of this.
Awesome. Ask good questions. And yeah, what advice would, and if Judy, you have a follow up, you can add that as well, along with your answer to what advice would you give to folks at home interested in becoming scientists or getting more involved in science generally?
Yeah, and I think on both of these, I would say be really, right, ask a lot of questions and be really interested in listening. Because, like Melanie said, you have to devote some time to just learning each other's language. And, but science as a language as a whole, sometimes you have to be patient with it. Like, you can't assume that, oh, I'm going to take one class and everything's going to be revealed and it's going to be, like it takes some investment, but I do feel like the payoff's really there, and that's true with collaborations too. Like sometimes it takes quite a lot of meetings of like, hey, what do you mean by this? Like, what are you talking about there? And working through some of those before you can get to the really fruitful, impactful, like exciting stuff. And if you're not like, oh, I had no idea what they said in that 30 minutes, so I'm just gonna move on, like, or a class, like, oh no, I'm kind of done here, like, that you may miss out, I think, on some of the future. So I would say, right, like be willing to invest a little bit of time and energy into it. And then I do think there's a lot of really still fascinating questions to be answered. So hopefully some of you will be able to be the ones to answer those. And but right, it takes quite a lot of effort getting there too.
Yeah, I think the advice I would give, especially for younger students who are, say college students or students thinking about going into college, and this, often science, majoring in biology or physics or scientific computer science, right? There's a lot of slogging through it, and I think an important thing is you've got to also keep your mind on like what's an exciting question. What do you want to do with this? I think that's one of the complex systems sort of lessons for understanding how things work is it's not just the top-down view. You don't just need the big picture. You also need sort of the bottom-up, the details of how things work. How the individual T cell moves through the tissue, in addition to like the emergent property of like, how many, what you can measure in someone's blood or viral load or something. Like you sort of need to keep your eye on both of those things at the same time. So in advancing a scientific career, I think it's really important that as you've got to simultaneously kind of slog through and learn the math and memorize the terms and really understand sort of the lower level pieces, which for me are the hard part. Some people like that. But also keep those bigger questions in mind. Like why am I learning this? Why do I actually have to understand this about the runtime of algorithms? Why do I have to understand, how to, the nitty-gritty details about, the receptors on this cell? All of those kinds of things, like you're really trying to piece together enough of the component knowledge to be able to answer a big question. And so I think a lot of people are either discouraged by the amount of sort of slogging through the details that's needed, or they get lost in those details and forget that actually it was for a bigger goal, right? And so that to me I think is important, and doing some sort of internship or research project or something where you're in a lab, or you're working with a team of people that's actually trying to solve some interesting problem. That's sort of where the rubber hits the road. So I would encourage that very strongly.
That's where you can see all the way from the ant on the ground to the big questions it's answering.
Absolutely.
Awesome. Well, thank you both so much for taking the time to share these stories. Tune into Proteus TV next week, everybody. Not next week, 2 weeks. Thanks.
Bye-bye.